fermentation it's a really like cool branch of agriculture. And I don't think people see it as agriculture because you don't do it with in an open field with a tractor.
Seems more like chemical engineering to me.
And I mean, you could say that, farming is chemical engineering too. You got to figure out, you have the right fertilizer. You've got to, you need to know your soil chemistry. You need to understand what you're spraying on your crops. Mm-hmm. So fermentation is just like the over-engineered version of agriculture, hyper-optimized by teams and teams of engineers and scientists, where you are trying to squeeze every last drop out of your inputs, your resources. So this just makes it super efficient. And when you're working with a fermentation vessel, it's a vessel, right?
Yeah.
It only has an outlet when you open that outlet. Okay. Versus like a cow or a cornfield, right? They you water a cornfield. Where's the water go?
Yeah.
It hits the soil. Some gets into the plant, and the rest goes downhill.
Yeah,
right?
Runoff, evaporations, tons of
runoff everything. With a cow, you feed a cow, and then it goes and it runs around. It passes gas. It does whatever cows do. Mm-hmm. It's not in a fixed location with every single input and output controlled.
Yeah.
So a fermentation vessel has that. You can have it as sophisticated as you want it to be, and that means you get to control exactly what goes in, and you get to control exactly what happens inside, and therefore what's going to come out. And any good fermentation system is going to completely utilize all of your inputs. 100% of your inputs are going to be converted into your desired outputs. There's always going to be some sort of like byproduct or waste product, and that's where you start getting like, oh, well, you can do certain things with like for example, like with yeast cultures in a brewery, right? The cell mass of the yeast is the waste product, right?
Yeah.
You need the yeast to make the alcohol, but the yeast itself is a waste product, and you can like give that to animals for feed to increase like protein content in like chicken feed, or you can just like send it to a anaerobic digester, and it gets broken down into like a fertilizer. So there's always something that you can get out of those outputs. But the point is that you're not wasting anything. You're utilizing everything to its fullest potential in a properly optimized system. And when we start looking at the issues we have with food in the current day, we have issues. We've got two issues that kind of seem to come to the front of my mind. One is lack of protein production, right, when it comes to crops. And the second is just general inefficiencies. So we're really good at making carbohydrates, right? We're really good at growing corn and wheat and grasses, right? Things that just make sugar and carbohydrates. We're really good at growing those. Those things grow like crazy. We're good at growing, protein crops too, like beans, like soybean is probably the biggest one out there. But we still use most of those crops to feed animals to get our protein, right? We're not growing the protein directly because it's really hard to get enough of it. If you want enough protein to feed a planet, you're not really going to be able to do that through growing beans, right? You got to grow a bunch of stuff and feed it to cows, and then you got enough protein for people. So now with all the issues with climate change, and we're trying to figure out, okay, how can we feed a growing planet without, ruining the environment at the same time?
Yeah.
And all the roads kind of point towards fermentation because it is so hyper efficient, and you get exactly what you want out of it. The byproducts are so few, and the efficiencies are so high, it makes perfect sense. So this new like holy grail that people are trying to find is, how do you grow a cow in a fermentation tank?
Yeah.
or how do you grow a tuna or a salmon in a fermentation tank? And it's really hard to do.
Because right? I mean, talking about what you were talking about before, like the losses. I mean, you're really, your inputs and your outputs are pretty in line with one another. So you're really and it seems like you can take, like I was talking about before, where you can take products that you weren't targeting necessarily, but then you can sell that, and then you're not limiting yourself so much. It feels like.
Yeah, exactly.
So this is kind of like the lab grown meat thing that you were highlighting here, and this is like a techno-economic analysis. So I guess what techno-economic is, that like they're analyzing and breaking down the food costs, like.
Yeah, techno-economic essentially means, so TEA is the abbreviation you'll see with it too. Essentially saying what's it going to cost me? You know, what are the economics of this technology? So if I were to deploy this at a commercial scale, because usually you have to like do a small scale and then you just model it at a large scale and you say, okay, we scaled this up to massive scales. It's going to cost me, what do they say? $13 per kilogram, right?
What's that? I mean, can you put that in perspective? Like how is that compared to? It seems cheap per kilogram, and I mean that seems very cheap for meat. I mean, especially
yeah, there's certainly like plenty cuts of meat that are more than that, right?
Yeah.
So with 2 pounds in a kilogram, so that's like $7 to $15 per pound.
Mm-hmm.
For meat. Like if I find red meat for $7 a pound at the store, I'm jumping on it. That's pretty cheap.
Yeah, yeah, exactly. Yeah, that's gnarly. So I guess here is what's new according to Elliot Schwartz. Previous TEAs looked at stirred tank reactors in the range of 10 to 20 kiloliters. Is that pretty large?
Mm-hmm. Yeah, that's pretty large. Okay. That's not. It's not like the largest. That's like mid scale. Like mm-hmm. a 20,000 reactor would be like, that's like kind of at like the pilot scale, maybe one step above that. So like lab scale stuff where you're first figuring it all out goes up to like, 20, maybe 50 liters, mm-hmm. depending on the lab. Once you start going to like the hundred to thousands of liters, you're doing pilot production. You're making serious amounts of stuff.
Okay.
But I mean, like for the largest things out there are hundreds of thousands of liters.
Wow. Jeez. That's gnarly.
I mean, go back to that other one. They even mentioned that in that same tweet, right? Much larger reactors, the 210,000 up to 260,
Yeah.
So, and they're talking about, so they have a couple key words in there, kind of go back to what I was mentioning before. So they say stirred tank, right?
Oxygenated.
A stirred tank reactor, oxygenated, right? Okay. These are all oxygenated because the main product is cell mass, right? They're not trying to make like a specific chemical or protein or something. They want to just make cells. And when you want to make cells, you use sugar plus oxygen, right? Combine those together and you get to make more and more cells. Mm-hmm. Versus, again, like classic beer fermentation, you take yeast plus sugar in the absence of oxygen and it makes alcohol without making any more yeast cells, right? Mm-hmm. So that's why, like, in a beer fermentation tank, the alcohol concentration goes up and up and up, and your yeast cell concentration stays flat or even like starts dying off and goes down.
Makes sense.
So for food production, if we want to make future foods, we want to be making cell mass. We just want to make huge quantities of cell mass. So you need to have it aerated. So stirred tank is literally like, I want people to think of like a blender, right? Inside of a steel tank, just like whipping around like crazy, chopping small bubbles into even smaller bubbles. And you've got like a big tube of air just coming in and it's just like blending that air up.
Wow.
And then they had on the same right there, they have go back, sorry, it was in that second paragraph. They have an airlift reactor.
Yeah, what's the airlift reactor?
Yeah. Yeah. So when you get to this large of scale, when you start getting to the hundred of thousands of liters of scale, the physics change, and you can't mix it anymore because you have like think about like something like a paddle, like a canoe paddle, right? And you're like spinning that on an axis, trying to like mix something, right?
Mm-hmm.
Now increase the volume that you're mixing by 10. Okay.
Mm-hmm.
So now you have to increase the size of your paddle by 10 or even more to get the same mixing mechanics that you got at that smaller scale. So your process essentially looks identical. And what happens is either you can't do it, or the energy cost becomes exorbitantly high because now the amount of like water pressure you have against your paddle
is increasing.
It's huge. You're mixing a paddle that's massive, right? It's the size of a car versus the size of a canoe paddle, right? If you were to scale it up accordingly. So all of a sudden you can't do these mixers anymore. You have to do what's called an airlift, which is essentially just a huge bubble column. So instead of having like a single inlet of air at the bottom that gets mixed above
the whole floor of the fermenter,
you just have the whole floor is just this big thing of air that's just like rub, 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 and it's just like going up and up and up. And these things are like 5-story tall buildings, reactors.
Wow.
They're massive, and they're just like huge. Yeah, and sometimes they'll put them in the ground too. It's like there's all different types of like systems that you can, or like designs you can use. But when you get this big, you can't mix it. It's too big. You can't. It's like mixing the ocean, right? You can't do it. So you just like bubble in a ton of air at the bottom. It's called an airlift or a bubble lift reactor, and it's just like literally a column of like a lot of bubbles, like just tons of bubbles, and it has its own like engineering challenges that come along with it. So
yeah, like Willy Wonka has shown, it's
exactly it's just like that. It's just like it.
I mean, how much volume in the fermenter is the air taking up?
That's I don't know the answer to that question, but I bet it is like in those big bubble reactors. I wouldn't be surprised if it was close to like a third or half, maybe.
Wow.
I really don't know the answer.
That's vigorous.
That's totally speculation.
Yeah,
but,
yeah.
I mean, you need to get so much air into those, like it's insane. And then you have there's all sorts of other complications that come up, like all of a sudden you have tons of foaming because you've got like this protein slurry that you're bubbling, right? So picture like the head of a beer. You're going to get that just building up and building up and building up, and all of a sudden your whole reactor explodes. You know, it happens all the time. It explodes, it overflows, your culture crashes, get contaminated because of that. So there's just like tons of challenges, and like I was saying before, getting oxygen into water, dissolved into water, is extremely difficult. It is not a reaction that happens easily. So you either need to have like a really, really high flow of oxygen into the water, or you need to have like really, really, really small bubbles so they have a high surface area to volume. The oxygen can then diffuse more quickly. So this kind of like a little bit of a balancing act that comes along with it.
So the gist is we're talking about big reactors. I mean, this is what the point, the 250,000 liter reactors, vessels. I mean,
picture like a building. That reactor is the size of a building.
Wow.
You know.
How many? That's okay. The pictures is not going to be for scale, I guess. Like, actually, why not? 200K liter fermenter.
Yeah, they'll probably just show you the top of them. You'll probably see like a dome top.
Yeah, I mean, it's hard to kind of see. Yeah, that's okay. All right. So, wow. So, okay. So here are the assumptions now that they are talking about. Let's see here. So the study first assumes a market size of 100 kilotons annually. Each facility is modeled to contain 50 of the largest production reactors. Wow, so there's your huge vessels, and now we're talking about 50 of them. The resulting total output of each facility is shown here, compared to the facilities modeled in other studies. So these are so these are the different papers, and we're talking about specifically Negulescu. So this is Negulescu on the far right now.
Okay. That's that's the author's name? Is that
this is exactly, Negulescu is the TEA. So I guess they're comparing these to the key assumptions of the different facilities. So this is, how do we know what the market? I guess this is cultured meat. So what is
meat? The market is meat.
But is that like a city? Is there like a moderately sized city or a town, or do you know what I mean? Like how does that relate to the market size of the entire market? I'm assuming probably less than 1%. Market size set at 100 kilotons annually. I guess, is that the entire market size, or just the market size for this that they would sell to? 25 kilotons for the airlift facility in batch process.
It sounds like it's their that's their estimated output. For this model, if they made that much.
That makes sense. Each facility is modeled at 50 of the largest. Okay. Jeez.
Yeah. So with those 50 reactors, they could make that much, and then they break down the costs for it.
Oh, this is gonna be hard to capture real quick. 1 second here.
I think the more the important thing that they're really trying to show here is with the lab scale data they have, they're starting to see for the first time modeled data that looks similar in terms of pricing to animal-based protein products. So it's not just science fiction. It's you're starting to see, and granted, these models, they can kind of be flexed a little bit, and that's why I think it's great that they're including multiple analyses here, not just one person. This is kind of like a review of multiple models, right?
Yeah.
And different technologies. So it's just starting to show that this stuff is possible. It's not total science fiction. And I really think that it is going to be coming in as soon as people are able to make consistent breakthroughs and actually get productivities. Modeling a productivity at that scale is one thing. Actually pulling it off is another 10 years and $100 billion. It's not going to be an easy thing to do. There's a lot of difficult hurdles that they need to overcome. But I believe that market pressures will definitely push it that way, and it will be overcome eventually.
Exactly what you're talking about. Since they lack impellers, the airlift reactors are so much cheaper. The airlift facility in this study costs roughly the same as the one modeled in Humbird, yet outputs 3.66 times more cultivated meat. That's incredible. Yeah,
they're a lot cheaper because you're not paying for that energy cost of the impeller, right? Running those things is super expensive. It's a massive motor that you have to run constantly. There's a lot of resistance on it, so just the energy cost alone is really high. There's other biological advantages to using those airlift reactors. So there's less contamination associated with them.
Interesting.
So
it's funny,
just because I think you have more air being pushed up into it, and I guess keeping stuff out of it at a higher rate. I guess you could also say If your air filter is garbage, you might have issues because you're pumping more air into it at the same time. So
it's funny, that's a kind of, I mean, I was one of my first chemistry teachers, whenever I was like mixing stuff, for a chemistry lab and stuff, I would like shake things up, but then he was like, no, you just like invert it and let the bubbles do the work. You just do this and just let the bubbles like mix it, and you don't have to like shake it. And it was just like, oh, dang. So kind of like in that same way, you're just constantly letting the bubbles like through the natural aeration, so letting the bubbles kind of do all the hard lifting there. Data from CM manufacturers on the grade of steel used for bioreactors in their facilities will improve the accuracy of future TEAs. Figuring out whether animal cells can actually grow in airlift reactors is another important data point to gather.
Yeah.
Ooh, animal cells, because that's I mean, we were talking about that earlier. It's very, I guess animal cells can, they're not talking about yeast there, though. They're talking about
We're talking about like cow or fish or pork, like any of their targets, right?
Because it's not going to have that cell wall like we were talking about, so it's going to be easier to shear the cell.
Exactly. It's not going to have a cell wall, so it's easier to shear it. There could be like viruses that get into your culture and kill it. There's a lot of challenges that come along with mammalian cells, like the media composition. I've never done mammalian cell culture, but I know that the media costs can be really high. Essentially, you have to like think about like what's in your blood, right? Yeah. Whatever's in your blood is like what makes your cells happy, right? If you're a healthy person, right, you got good blood, like your cells are going to be happy and you're going to grow, and that's what you need to mimic. So like blood is pretty close to the type of media that you need to make for any mammalian cell, and that's not a simple media. It is complex. There's like, it's not just like salt ingredient. It's not just like, oh, I need more nitrogen, It's like, no, you're missing like a protein or a hormone or a stimulant, and that just makes the control factor so much more difficult, so that's outside my area of expertise, but that's where all these companies are fixated on. It's understanding how do we make not only like the right media, but an affordable media. That's the thing, because it's really easy. Like there's mammalian cell culture media is available commercially. You can go buy it and it works great. Yeah. But it's prohibitively expensive. So you have to make like some cheaper synthetic form of it, right? Mm-hmm. And then the other difficult portion of it is like getting these mammalian cells to not burst open and die, or get them to actually grow. And then the third issue is then you got to make it look like the animal when you're done with it, right? Yeah, exactly. Because it's going to be, what's it going to look like when it comes out of the reactor? It's probably going to look like a bag of blood. It's probably going to look like it's probably going to be it's not going to come out mooing, looking like a cow, right? Yeah. It's going to be like if you took a cow and put it in a blender, and now you got to like turn it into steak somehow.
Yeah, it's a tough problem, man. It's an interesting, like, how I've seen like 3D printing of like taking some of these cells and then using like 3D printers to kind of like arrange them. I've seen other strategies and stuff in order to do that, but that seems like another like structural engineering problem entirely.
Yeah, I think the terminology is like scaffolding.
Oh, okay, like, there it is.
What's going to be like your product's scaffolding, because you have to like have these cells. I think it's like the same problem for like growing organs for transplant, right? Yeah. That's like another area emerging in the medical field that kind of has like some similarities to it, where like you can take somebody's stem cells and you take them and you go and grow them a new heart, right? Mm-hmm. Like that's like what we're progressing to. Like pretty soon, you're going to be able to grow your own organs as your own transplant. You don't need to have someone else donate an organ to you. You can go grow your own pretty soon, right? But amazing. You need to have like, I mean, they have it. They're getting there. You know, they've grown like cartilage pieces, like they've grown like ears and noses and stuff. Like, you know, but you need to have like that like scaffolding that the cells attach to. Like they're not going to go into the shape on their own. But if you can get them into that shape, all of a sudden they like might assume that function or something. You know?
Interesting. Yeah, I guess. Oh, man, that's so hard to think about because I'm a microbiologist, so thinking that just about how they would, what do they evolve to do? You know, like how I give them a similar scenario. How would they assume that? It's kind of like if you heat up like bent steel, sometimes it'll like go back to the learned shape.
Right. Yeah.
Like,
yeah.
How could we like mimic that like evolutionary drive?
Right. Just like shock them. It's like they just jump back into place.
It's very weird. OPEX, I guess that's operating expenses.
Yeah, that's operational expenses versus like CapEx would be like your construction costs.
Gotcha. So operating these things now, costs are less interesting and largely dependent on the bioreactors themselves. Bigger bioreactors, fewer facilities, and lower labor and facilities overhead costs. So I guess that makes sense but then it's like, you better get that right because if you get one of these viruses or one of the tanks crashes, that's like, you're done.
Right.
So kind of,
right.
So summary table below, oh, this is going to be really tough. Let's see here. Yeah, this is a cool part of, I guess, microbiology, fermentation. I mean, you'd normally think that it was, but man, it feels different. It feels.
It's a lot of microbiology. It's a lot of chemical engineering. It's a good bit of physics. There's a lot of. That's why I love like working in that space. It's like the intersection of like all these like really cool areas of science, and they all have to work together. And it's like, you know, you have to understand all of them to be good at that fermentation process. You can't just be a good chemical engineer. You also need to know the microbiology. You know, you need to understand how, like, you know, what does your cell need? What can your reactor do? It's like constant feedback, you know, of information and data. And like, it's not just like one team is going to solve the problem. It takes like this multifaceted approach to figure these things out. So all these startup companies that are in this space, you know, it's just like, I think a couple of them are going to do really well, you know. Just a lot of exciting stuff going on there.
This is interesting. There seems to be a crazy point where the cost to build these, I guess the STR is the one with the propellers and that kind of thing.
Yeah, that's stirred tank reactor.
Stirred tank. Okay. I mean, that's crazy expensive. I mean, compared to the airlift that we were talking about earlier. I mean, the airlift, the 260K is cheaper than the.
What size? Yeah, what size is this? Oh, yeah, the top there, the 210, 260.
Yeah. Yeah. I mean, look at that. Look at that savings in costs. And then even the operating costs, it's much cheaper. These airlifts seem to be. I wonder what the capex is, or the cost for construction is so much higher for these stirred tanks. I wonder if it's just like the propeller, all the propellers, all the tubes going in, and there must be additional type of regulatory things like where you have. You know, on the air tanks coming in kind of thing, but you have to have them all regulated. You have to have them all probably filtered, obviously, so that they don't have their own contamination issues, as opposed to just one airlift bottom floor kind of thing.
I mean, the airlift ones still have like all the other controls and sophistications. It's really, the only difference really is like having that impeller.
Wow, that's crazy.
So it must be that expensive to have that. There's also like, I don't think this would affect the cost very much, but like inside the tank, they'll have baffles, right? So it's like picture like a tank, and then on the inside you have like these fins that are fixed against the wall. Yeah. And then in the middle, you have your impeller that's rotating against it. So you have like a fixed blade and then a moving blade, and those are going past each other, and that just creates like extra turbulence and stuff. But I can't imagine that would actually cost much.
What's COGS stand for? Cost of
goods,
goods and services. Okay, with depreciation or without depreciation, much cheaper. Just this airlift thing is really interesting.
You know,
it's really highlighting it.
I think there's it's really difficult to do airlift at small scale. It doesn't work at small scale. It almost like only works at really big scale. Because at small scale, the size of the vessel, like picture like lab scale, right? Mm-hmm. 1 liter, a 1 liter vessel, and all you're doing is bubbling air up through it. You're not going to get the same effect as if you're bubbling like a huge volume of air through this massive tall column, because the retention time for that air is just like, you get a lot more time for the air to dissolve into the water in your larger vessels. Mm-hmm. Where in the small vessel, you're just going to like evaporate your culture, if you could bubble tons of air through it. So that's why like this doesn't really have anything to do with like, which organism you're growing. This is more just like the difficulties of taking something from a lab scale to a commercial scale. It's like, you're going to do most of your research in a stirred tank because that's what works really well at laboratory scale. And then you have to transition that process to a larger stirred tank, and then you have to transition that to an airlift reactor. Like all of a sudden you change your variables when you're going through the scale up process. And it's like, thankfully it's pretty well established for, there's a lot of examples out there of how to properly do it. But like, for a new organism, there's always going to be new challenges, and you never really know what's going to happen until you go and do it yourself.
Yeah, actually happens. Yeah, and it seems like a lot of these new meats and stuff, they're going to be a funneling effect where it's like, we don't have fish for a while, guys. We haven't figured that out yet. You got beef. We got beef. Sorry, that's you know what I mean. It's like
there's a lot of. I know a few fish companies, and there's definitely cow. I don't know anybody that's trying to do like chicken or pork. I'm sure somebody's trying to do it. Mm-hmm. I just personally haven't looked at it or come across it. But yeah, I wonder what's going to be like the first one. And I think right now, only in like Singapore is this stuff legal to eat. I think they might be the
that's
only
a good point. I mean, the FDA is so conservative in the United States for like letting things through. It's very strict and very hard to get things through.
The FDA adds like a whole, and like, I mean, it's strict in the U.S. and that's even more strict in Europe.
Oh yeah.
So like they can't even eat like any GMO food in Europe. Yeah.
I mean, I guess like, they would have to be genetically modified, wouldn't they? The product that they're growing. I mean, that's how else are they going to genetically engineer it in order to make this certain product?
I don't think it has to be. Wow. I think it's more about the fermentation process and just like process engineering. I don't think it's about genetic engineering. And honestly, I think that like if I were to start a company like that, I wouldn't want my product to be genetically engineered because it's only going to make it that much harder to sell it, right? Like not only is it already a cell-based meat product, but now it's a genetically engineered cell-based meat. Like, come on, people are going to get scared away by that. Like,
oh man, that's crazy.
Like,
I mean,
we can do mammalian cell culture. You can just like take a mammalian cell line and you can put it in media and it'll grow without being genetically engineered. You could genetically engineer it to like make cooler stuff, but if all you're trying to do is make the original cell, like I want to make more tuna cells. I don't want to make anything special with it. I just want more tuna cells that they naturally exist. That's not a genetics problem. That's an engineering problem. That's media optimization. That's process design. That's like understanding how to do cultivation and fermentation of these things. But I think the genetics can be left alone.
Interesting. That blows my mind. Yeah, that's I guess especially if you want the broadest market possible, like why limit yourself kind of thing. Even if you can make it 200% better, the product, the yields, then if you're limiting yourself and your market by billions of people with Europe and China and the United States, then there's no way that it's a better. So at these costs, cultivated meat is within range of premium products, but still out of range of most beef, pork, and chicken. One way to lower costs is developing hybrid products with costs diluted by cheaper plant-based ingredients.
That could work. Like instead of doing, I think it's Beyond. They use like beef juice to make their stuff red. Mm-hmm. Like what if you just replace beet juice with cow cells?
I mean, wouldn't that be more expensive? Cow cells? I guess if you have blood. Well, I guess if you have blood.
It would be more expensive than beet juice. Yeah.
Yeah.
But it would be, you're not trying to sell a whole quarter pounder patty made of cow cells. You're only selling
the blood.
You're only selling a gram of it. Well, no, I'm saying from the cell-based cultured cow cells, right? You grow them in a fermentation tank, and you put 1 ounce into that 16-ounce patty that you're selling, whatever it is, right? So it's 10% of it versus being 100% of it.
Gotcha.
And your costs would be more manageable that way.
Interesting. Wow, this is blowing my mind. This is cool. The authors of this study show the media optimization is another path, kind of like exactly what we were talking about. Using the price to volume regression equations in Humbird, the extrapolated medium formulation costs modeled in the study work out to $1 a liter.
That's great. That's cheap. It could be cheaper, but that's pretty cheap.
Wow. The components other than glucose, amino acids, and proteins were collectively found to consist of a mere 0.17%. That's all those hormones and all those things that you were talking about that kind of made it so expensive. So they were left out of the model.
Yeah, the glucose and the amino acids, those are going to be very expensive just because you need, not because they're expensive per weight, but you just need a lot of them.
I wonder what kind of, I mean, even if they were only 0.17% of the total media. Oh, I guess, no, I'm sorry. It's the total media cost. I thought that they were talking about the total media amount. So they're talking about the cost of it. That's amazing. $1 a liter? That seems cheap. That seems very cheap to me.
Yeah, I don't know. I don't know what type of cell densities they're getting to, but in the grand scheme of fermentation, if you can get to 100 grams of cell mass per liter, you're doing pretty good. So you would be essentially 100 grams of product for $1 would be $1 media cost outside of the main cost bill. The fact they didn't include glucose in their costs for the media is I don't know, kind of nonsense, because glucose is your main cost for media. It's you're putting more of that in than anything.
Interesting. Yeah, because it's it's just interesting to compare it to the costs compared to actually making, the cow and stuff like that. Like what does the cow need versus what does this need? Like what kind of,
Right.
nutrients that you're going for. And it seems like that's pretty cheap for.
I always love seeing how many, how many gallons of water does it take to make a hamburger? It's like a couple thousand gallons of water per hamburger.
Yeah, exactly.
Not per cow, per hamburger, right?
Mm-hmm.
It's like, I forget, I think it's like 2,500 gallons. It's just like something ridiculous.
It's insanely expensive.
Yeah,
because you have to water the plants. You have to water the plants. That's the food for the cow, and then the cow drinks water.
And then the cow drinks water, and then you gotta wash. Then you gotta wash the meat as you're processing it. It's through the whole process of it. It's not just what the cow drinks.
Yeah, exactly. But this is cool. So this is kind of like what he was talking about here. Then we're fairly confident that switching to food grade media ingredients and coming up with clever innovations for growth factors can allow lower media costs substantially, with some modeling costs as low as $0.25 per liter, which would be a fourth of the cost that this study is showing. That's impressive.
Mm-hmm. Mm-hmm.
So modeling by this new paper suggests that at these media costs, the cultivated meat product in a food grade airlift reactor, which is the larger super grade, could cost between $5 a kilogram and $15 a kilogram in a large stirred tank. Wow. So it's way cheaper.
That'd be way cheaper. It might be it's a pretty optimistic model. Does that include the glucose cost though in that final assessment there? It has to, if they're going to say that.
COGS with depreciation versus media costs. Looks like it. Looks like it.
So I wonder what I wonder what, like, since these are like new cell lines, these have like really never been grown before.
Mm-hmm.
So I'm curious what their conversion rates. So like what's called the yield is. So like the yield when we're like talking like fermentation tanks and stuff like this, yield refers to how much do you get out from what you put in, specific to like your carbon substrate. So if you put in a kilogram of glucose, are you going to get half a kilogram of cell mass out of that culture? Or are you going to get, a tenth of a kilogram?
It looks like the media cost drivers are shown and it has glucose in it. This is fairly well known already. Our most recent call for grant funding aims to find lower cost and non-recombinant alternatives. So exactly, non-GMO is basically what he's saying, non-recombinant alternatives for transferrin and albumin. You can see why we're targeting these proteins for the pie chart.
Transferrin's a super expensive part of the media there, because that's like it's made recombinantly. So if we're gonna do it without our recombinant, I don't know how they're gonna pull that off, but that's what helps get oxygen into the cell.
Mm-hmm. I mean, albumin is the number 18. It's 18% of the media. I don't know why this isn't showing up well here. Here, 18% of the media, or $25 per gram. That's a lot of the cost.
25 cents.
25. Yeah, sorry. Thank you. 25 cents per gram. And glucose and insulin. That makes sense. Wow, that's a. I mean, there's so. I was just thinking about the stocks and stuff for all the corn and stuff, and but I guess they're already taking the glucose out, which is why they're using it for yeast and yeast food to make biofuels and stuff. So there's not going to be anything there, but there seems to be. Man, if there could be like a food waste or something, glucose, like all the processed and sugary foods that people throw away constantly, that seems to be the like a good way to get that stuff. I don't know. we have so much food going into our landfills right now, and then you look at the cost of this, and you're like, dang, the glucose is a huge portion of this. If there is some way to recycle that food cost.
I mean, glucose is like extremely abundant, though. At the same time, you know, it's like it's not hard to get a couple hundred tons of glucose if you needed it, right? Because it's just glucose.
Yeah.
Everybody uses it.
Yeah. What are the cheapest sources of glucose right now? I guess. I guess they're calling for grants for even lower cost, non-recombinant.
Yeah.
So I'm assuming it's recombinant.
I don't know. I think like whatever's closest is probably going to be cheapest. I bet transportation. Glucose is so cheap. I bet that transportation costs probably play a big role in what you're going to buy. You know.
Good point.
Like it might like you know, corn, sugarcane might be cheaper than corn, but you're not going to get sugarcane in the U.S. You're going to get corn.
Yeah.
Like, are you going to ship sugarcane from Brazil or something, or are you just going to buy corn glucose because it's right there? So,
yeah.
you always need, like, whenever you're doing these things, you need so much of it that it's not like you need like a constant supply. It needs to be nearby. So I think your location is going to determine what you use more so than anything.
I mean, what about? I mean,
you're really large.
We were talking offline about like how productive algae ponds are compared to land plants. So I guess like why not? Could somehow like algae ponds or something like that be used to make all this fuel? We say glucose, but really it's fuel for these other kind of things going forward. Yeah. I know there's plenty of people that are looking into doing that. Essentially just taking like a high carbohydrate concentration algae strain and growing that and then just fermenting it, use it like they'll do something like they'll just, make a cell lysate and then that is the media, for your host. So there's I think there's a lot of potential there. I think that is still hung up on like just the general costs of like growing the algae. There's a lot of expenses that are associated with that. It's so hard to compete with like traditional agriculture, corn and soybeans. It's already there. I mean, infrastructure is already there. Like you were talking, the train's already right next to it. They're already kind of like set up. They already have the autonomous vehicles or farm equipment now. So kind of like switching over to liquid-based algae ponds seems kind of like just another world away kind of thing. Right.
Yeah, maybe. Yeah, give it another like 50 or 100 years and maybe out of necessity it'll happen. Mm-hmm. But yeah, I mean it is way more productive in terms of like how much you get out of an acre of land, but
that CapEx cost is kind of creeping up on you.
The CapEx cost is really prohibitive. Yeah, for something that's like you have to build, because like those like algae ponds, they're really similar to like building a fermentation plant, except it's it's an open system on its side versus like a big steel vat standing up.
Yeah.
No, they're expensive.
Well, maybe that's something like we'll see, kind of like a fermentation algae pond hybrid kind of thing, where you have like your algae solar panels making the sugars that need to be fermented to make these cultured meats kind of thing.
I mean, that's the thing is like the glucose is always going to come from an organism. It's not like you're not making glucose out of petroleum, right?
Yeah.
You're making it out of organisms.
So in summary, this new TEA shows that bioreactor economies of scale will be important for lowering cultivated meat costs, and a pathway to cost competitive products could emerge. It's important not to overinterpret one study. This is a preprint and thus subject to change. That last part about preprints is very I was a little stipulative just to cover this, just because of like the preprint, but I thought it was a good Twitter thread by Elliot Swartz, so I wanted to highlight it. But I think that's man, that the economy of scale, especially with the airlift, is just incredible. That's so cool.
Yeah, I mean, these models always need to be taken with like a fairly large grain of salt. There's just like you're taking data from like probably I don't know what they said in there, but it's probably like 1% the volume that they're projecting to, and extrapolating out very far from where they are right now.
Yeah.
But there's some data behind it. It's not make believe anymore. So
it's very cool. It goes into the assumptions here. So each study has its own assumptions. Notably, in this study, a metabolically efficient cell is assumed. So that's kind of a stipulation. Mm-hmm. Oxygen transfer assumptions need validation, and assumed media sterilization and contamination events are very favorable. So that's what we were talking about, like with like the viruses or something like taking over and crushing the entire thing.
Literally just like bacteria or something getting into your culture. Yeah.
I mean, these are. I mean, I'm assuming the doubling time is atrociously bad, so you're going to be outcompeted by bacteria.
It's probably got yeah. Like, is it going to be like days or something? Like how? Like, I have no sense of how quickly these cells grow.
Yeah, and how long are these? How long are these cultures going for? I mean, are we keeping it at exponential growth phase kind of thing where we're constantly fed batching it? I don't know if they do.
They have to be. It has to. Okay. They have to do a fed batch. But but I mean, like, bacteria's got a doubling time of like hours for slow bacteria. Like,
yeah, exactly.
And mammalian cells, I don't think grow very quickly. So they're going to have to, and I think that's why maybe they're focused a lot on the media, because that's going to control how quickly, because that's the immediate environment that they're in, right? Whenever they're floating around, and that's going to control the underlying phenotype and like how quickly they're actually going to grow and divide. So I really actually never actually looked at or seen fermentation like growth curves and data from those companies yet. I don't know if they're published. They might not want to publish them. It might look pretty bad.
So there's something that you forgot to include, that according to calculations by Humbird, reactors greater than 20,000 liters will run into problems with carbon dioxide inhibition. Thus, this is also an area to further dissect in future modeling of large reactors. Bioreactors greater than 20 meters cubed have a lower maximum cell density due to CO2 inhibition, while
interesting.
Ammonia inhibition can be mitigated.
Sorry, go ahead.
So that's not ammonia. That's NH3 is. I thought maybe it is. I don't know. While NH3 inhibition can be mitigated to a degree with enhanced metabolic efficiency, CO2 inhibition cannot. Regardless of the metabolic parameters, the respiratory quotient, CO2 over O2, of the growth reaction will remain around 1, and the CO2 stripping rate will remain equal to or less than the O2 transfer rate. The only way to circumvent CO2 inhibition in a sparge bioreactor is to sparge harder, possibly to the point of cell death. So there's that. Sparge is the getting that new air in there. Yeah, this may preclude the scale up of animal culture into extremely large bioreactors.
Interesting. Yeah. So essentially, what they're saying is that when you're doing
ammonia, NH3 is ammonia.
When you're doing aerobic fermentation, like these guys are, like the cell is taking that glucose plus the oxygen, it's making cell mass and giving off carbon dioxide. And that carbon dioxide dissolves into water a lot quicker than oxygen dissolves into water. So that's already difficult because now you have an imbalance, right?
Mm-hmm.
And you're going to be bubbling oxygen into the water, and some of that oxygen is going to strip out the carbon dioxide from the media, right? And that'll bubble out and diffuse. But since you're also feeding in tons of sugar at the same time, it's going to be constantly making CO2. So there's going to be a buildup of CO2 dissolved into that culture, which is clearly, like, I guess with these types of cell lines, that's an inhibition. And that's something that always happens when you get really high cell densities in any fermentation tank.
Mm-hmm.
Again, back to the beer example, when the alcohol concentration in a beer gets too high, the yeast cells start to die, and then they stop making alcohol, and your fermentation is over. So like every batch of beer kills itself, right?
Yeah.
The yeast will consume all the glucose to the point that they make, like all those really high alcohol beers, they need to have like specific high alcohol tolerant strains. Otherwise the yeast just die off at like 5 or 6% alcohol, and they can't make anymore. So there's always something that usually pops up in every fermentation culture that starts to inhibit. It might be like salt concentrations, might be like I just said, alcohol. In this case, apparently CO2 is the thing that they're going to be worried about. And then, so in order to get rid of it, you have to bubble even more air through the culture. And when you start bubbling more air into the culture, the bubbles themselves can shred the cells. The actual mammalian cells get ripped open through the shear stress of like all these bubbles going past the cell, and it'll just kill your culture. It'll rip the cells apart if you bubble it too vigorously. So it's going to be really difficult. It's going to be really difficult to pull it off. It's like they got to figure out a way to either get that CO2 out by a different mechanism, or invent a hardier cell line that doesn't rupture as easily, so they can bubble more air through.
Interesting. I mean, is the CO2, it's dissolving into the mixture as well, making carbonic acid and making it more acidic over time? So you're doing...
That's what I'm guessing is happening here. Like, we don't have all the details of what they're talking about, so I'm not sure. And I'm like, I don't work with mammalian cells, so there could also be some like signaling mechanism where like CO2 is high. And because like I know like with like in humans, like we have like a regulatory mechanism inside of our bodies that is controlled by CO2 levels, right?
Yeah.
And carbonic anhydrases, and like it controls like our heart rate and something like that. I forget. I don't know. I'm not a doctor. But so it could be that. It could also just be like a pH thing. It could just be like the culture is getting too acidic and the culture is dying off, which would be like if CO2 is building up, 100% it's going to be dissolving into the media. And because like think of it, the cell is in the media, the cell is pushing CO2. It doesn't immediately go to the headspace. It's got to go through that whole culture first, right? So it's going to dissolve, and then it'll bubble out at the top. But once it dissolves, it's going to make carbonic acid. It's going to lower the pH of the culture. The cells might not like that, and then they die.
Very cool. Very cool. Well, that was really sweet. I appreciate science Twitter when it's good, and I love these like going over
that's a nice way to like. I haven't seen too many of those, but that's a good way to like have some public interaction with a publication like that. Just like pick out the highlights from a paper and put them on different tweets and everything.
Yeah. I've done. David Liu did that he does it with his papers. So I kind of went through my papers too. I've done that, just kind of like going through and summarizing each figure. You're kind of like doing a figure caption, but kind of like taking the big takeaways. And that was a really good. So follow Elliot Swartz. I appreciate all his.
Good Food Institute. They're like the leading group in this field right now in terms of. They don't actually do the research, but they like do a lot of like supporting roles for it.